Hello everyone. We have known immune organs, immunocytes, and immune molecules. In this section, we will talk about immune response. You may ask how the host defends against pathogen infection and how the immune response takes place. To make clear these questions, we are going to introduce the process of immune response and its major functions. Broadly, immune response includes innate immunity and adaptive immunity, barrier structures, innate immunocytes, and immune molecules contribute to innate immunity. Adaptive immunity includes T-cell mediated cellular immunity and B-cell mediated humoral immunity. Here we just introduce adaptive immunity. Adaptive immunity refers to the process that immunocompetent cells recognize the antigen specifically, then activate or lose their ability to be activated, proliferate, differentiate, and play immunological effects. Adaptive immunity can be classified into induction stage, reaction stage, effect stage. Now, let's look at T-cell mediated cellular immunity. Firstly, APC, uptake, process, and present antigens to T-cells. TCR on T-cells recognize antigen peptide and MHC complexes on APC specifically. During this process, CD1, CD2 recognize MHC alpha helix, and CD3 recognize antigen peptide, which is called dual recognition. The second stage is activation, proliferation, and differentiation of T-cell. T-cell activation requires dual signal. The first signal is a specific antigen signal. TCR recognizes peptide MHC complexes, and CD4 or CD8 enhances the first signal by binding to beta 2 or alpha 3 domain of MHC. The second signal is co-stimulatory signal, which is provided by CD28 and B4, CD2 and CD58, or other adhesion molecules. Resting T cells express low affinity interleukin-2 receptor. Activated T cells express high affinity interleukin-2 receptor and secrete cytokines such as interleukin-2. T cells proliferate and produce a lot of daughter cells aimed in interleukin-2 by autocrine or heterocrine. CD4 positive daughter cells differentiate into effector T cells, and some differentiate into memory T cells. CD8 positive daughter cells differentiate into effect CTL, and some differentiate into memory TC cells. Memory T cells are the basis of vaccination. T-active cells release interferon gamma to activate macrophage, mediated inflammatory reaction, and inhibit the functions of T2 cells. T-active cells release interleukin-2 to promote the proliferation, differentiation of T-active cells and TC cells. T2 cells release interleukin-4, 5, 6, 10 to activate B cell to produce antibody. T2 cells release interleukin-2, 5 to promote the differentiation and development of eosinophils and mast cell. T2 cells release interleukin-10 to inhibit the activation of macrophage and function of Th1 cells. More than 2 decades ago, helper T cells are viewed as having two major phases, Th1 and Th2 cells. But now, the new diversity of helper T cells is identified, including Th17, Th9, Th22, TFH, and different types of regulatory T cells. CTL, precursor, recognize peptide class 1 MHC to proliferate and differentiate to effect CTL at action of interleukin-2 released by Th1 cells. The effect CTL specifically recognize and bind antigen on target cells. CTL release perforin, granzyme, and express phospholagate to kill target cells. Cellular immunity plays an important role in defense against intracellular microbe infection. CTL can kill tumor cells or virus-infected cells directly. Cellular immunity is also related to graft rejection and graft versus host disease, GVHD, as well as type 4 hypersensitivity. B cells mediated immune response is chemoimmunity or antibody-mediated immunity, which is a total immunological reaction. Then the B cells recognize antigen, then activate, proliferate, differentiate into plasma cells, and produce antibody. The first stage is antigen recognition. PCR directly recognizes the epitope on the surface of antigen. Then B cells capture antigen by PCR, present antigen signal to T cells. B cell activation also requires dual signals. PCR recognizes determinants on the surface of antigen, and IgA transmit the first signal. CD19, CD21, and CD81 binds to C3DG on antigen and promotes PCR to recognize antigen. The second signal is provided by CD40 on B cells, binding to CD40 ligand on activated T-at cells. T-at cell and B cell are interacted. B cells act on T-at cells by presenting antigen to T-at cells, providing B7 for T-at cells. T-at cells act on B cells by providing co-stimulatory molecule for B cells, and secrete cytokines, help B cells proliferate and differentiate. Active B cells express receptors of cytokines such as interleukin-4 receptor and interleukin-5 receptor. Activated Th2 cells secrete cytokines such as interleukin-4 and interleukin-5 to enhance proliferation and differentiation of B cells. Proliferated B cells form germinal center, where B cells differentiate into plasma cells called antibody-forming cells to produce antibody, partially into memory B cells. In germinal center, during proliferation and differentiation, somatic hypermutation of B cells induce affinity maturation of antibody. Some B cells differentiate into the plasma cells, which produce IgM. Some B cells have an isotype switch to produce other types of immunoglobulin. Some B cells stop differentiation and become memory B cells. Memory B cells take part in secondary immune response. When the same antigen stimulates these memory B cells, a secondary immune response is elicited, leading to more rapid proliferation and differentiation, and production of great quantities of specific antibody that are produced in the primary response. The effect of B-cell-mediated immune response is performed by antibody. We have introduced the function of antibody, including neutralization, complement activation, opsonization, as well as ADCC and type 1 hypersensitivity. In this section, you should know the process and function of cellular immunity and humoral immunity. Thanks for your watching.